Hello, everyone, and welcome to phyloseminar.org. The current theme is on phylogenetic models for continuous traits and their applications. In this series of 3 talks, we're hearing 3 perspectives on continuous traits. The first 2 talks were from Josh Schraber and Joseph Ueda, and today Michael Landis will describe his work on pulsed models for trait evolution. If you want to ask a question, you can either tweet phyloseminar, or you can type your question in the live chat box to the right of the video on YouTube. As I mentioned, today's speaker is Michael Landis. Michael got his PhD in integrative biology with John Uhlenbeck at UC Berkeley. After that, he worked with Tracy Heath at Iowa State and is now a postdoc with Michael Donahue at Yale. Welcome, Michael, and thanks for participating.
Hey, ready to go? Can you hear me fine? Okay. Yeah, so my name is Michael Landis. Thanks for the nice introduction and for inviting me to speak today. I'm following up 2 very interesting talks that were given by Josh Schraber and Joseph Ueda. And my talk will be a little bit different in theme. Josh's talk focused very much on how we can apply models of continuous trait evolution to gene expression data sets. And Joseph talked about some of these methodological problems and sensitivities regarding what models we apply to the data itself. So I'll be speaking a little bit about how we conceive of these models of trait evolution and how they operate over deep timescales. When we're talking about how traits evolve between species, typically we're considering timescales on the orders of millions of years, the timescale that species originate on. And one of the many inconveniences that coincide with humans living short lives is that we can't really observe these changes in real time, and instead we have to make statements about how traits might evolve for the most part. So we see this variation at the present, and we know that some evolutionary force must be responsible for that variation, but what forces might be acting? And throughout the history of evolutionary theory, we've always relied on these descriptive models for how traits evolve over deep time. And we hope that these models resemble reality to some extent. And recent advances have let us actually test whether what we think is going on is a good fit for the model. Now, starting with Darwin, he placed a strong emphasis on the role of gradual change over long time scales, followed by Simpson, who introduced a conceptual framework for distinguishing between different aspects of evolution, including the tempo, how fast traits change over time versus the manner in which they change. And then with Eldridge and Gould and their theory of punctuated equilibrium, which in essence pushed back against the reigning paradigm of gradual evolution and argued that most evolutionary change is concentrated into relatively short periods of time. One of the reasons that Eldridge and Gould and many others argue that evolutionary change is concentrated in short periods of time into what I'm going to be calling evolutionary pulses is because of the fossil record. And normally when talking about trait evolution and variation, people like to show these pictures of really beautiful assortments of charismatic fauna. I'll show you something that's interesting, I think, instead of inherently beautiful, I guess. But here we have a group of different fossil bryozoans. And while they're perhaps not as beautiful as a lion to the public, to me they're really beautiful and interesting in their own way. And part of what's so cool about them is this story that they tell. When you look at their fossil record, this is a figure that's modified from a paper from the 80s, and it's one of the papers that was used as strong evidence for these models of punctuated equilibrium. And this figure shows that these fossil lineages tended to exhibit long periods of evolutionary stasis where almost no change happens over orders of millions of years. And one thing that quickly is troubling when you look at this figure is that when you have stasis maintained over long periods of time, we have variation between lineages. How do you reconcile these 2 facts? At some point, you need essentially these horizontal lines that reconcile the difference. And so when studying trait evolution, one of these fundamental problems is how do we reconcile the fact that variation was generated over time with the fact that the fossil record is largely dominated by stasis. And one of one of the working models that I'll discuss today is trying to separate microevolutionary versus macroevolutionary components. Researchers such as Eldridge and Gould argued that what we observe when we observe trait differences between species is dominated by some underlying macroevolutionary process, and the microevolutionary process is essentially just noise working on top of that. So here we have time on the x-axis and trait changes on the y-axis. And we see that if we change we see that if we have a stationary optimum, the red line, and we have a population evolving under stabilizing selection attracted to that optimum, we have no great amounts of variation arising over these long time scales. If you took the population at the beginning of this time, the population at the end of this time, the traits would be essentially within the same region of trait space. If you instead allow the optimum to change, say the adaptive optimum shifts over time using a sort of gradual random walk, then we see that variation can accrue, or if the adaptive optimum shifts rapidly due to, say, the appearance of new predators or the colonization of new areas, we can see that the population's trait will track that optimum and shift relatively rapidly. Applying models of this type, where you have an optimum that is changing with time or stationary with time, and a population whose traits adapt to that optimum, when applying these models to fossil time series, some paleontologists, Gene Hunt, for example, who's done a lot of great work in this area, have found that stasis is extremely common in the fossil record, not just in a sort of observational sense, but in a statistical sense. And on top of that, he's found that when you have sufficient data, many fossil series contain these pulses. It's not that all fossil series contain pulses, but many do. And so we think that through these types of observations, we should in some way account for these pulses when talking about how traits change over time. The simple model that the model that he proposed was a model with I think 5 parameters. We have a time of a shift, parameter describing the evolutionary stasis before the shift, a parameter describing the evolutionary stasis after the shift, and 2 parameters explaining the optima before and after the shift. Really nice model. It was primarily designed for fossil data, which makes it fairly easy to work with because of the immense amount of information you have. For another nice study in this region, studying pulse models of evolution, is by Yosufu Ueda and others. In 2011, they introduced a model where they allowed evolutionary bursts to arrive according to a Poisson process. And in their study, they found that for approximately 1 million years, very little divergence accrues between species. And after that period, you begin to see a burst of divergence. And this is, they termed this the million-year wait. And they found that this pulse model, where you have multiple bursts, best explains the data. The multi-burst model, if you compare different evolutionary models, the multi-burst model looks something like this, where you have, like I said, an optimum that remains relatively constant over time, and periodically the value of that optimum can shift. And when the optimum shifts, the population which is in stasis around that optimum tracks it very closely. And this model was designed primarily for large data sets composed of taxon pairs ranging from field study data, fossil time series, and some phylogenetic evidence. Now, when we're modeling these major features of macroevolution, typically the way that if we want to get some understanding of how these evolutionary modes operate over the tree of life, we begin with some descriptive model, like the ones that I introduced. We move then into defining those descriptive models as statistical models. Having a statistical model isn't enough. If we want to actually test whether these models are good descriptors of what we see in nature, we need these inference methods to essentially bridge that gap. One of the first steps difficulties in this process then is defining what a pulse is. And throughout this talk, when I'm talking about a pulse, I might use different words intermittently, but a pulse I'm defining, there's some synonyms that pop up in the literature include jumps, shifts, punctuation. And by pulse, I mean an event where the amount of change exceeds what's typical of previous intervals of similarly short length. And so perhaps a more mathematical way of thinking of that is when the dx dt is much larger than the average background effect. And various mechanisms have been proposed for pulses. One thing about defining pulses that's difficult is pulses are sort of defined relative to some other evolutionary mode. And most common it's juxtaposed against stasis. And often when describing mechanisms, there's a mechanism that is constraining how freely a trait may evolve, and then something that is relaxing that constraint. So one mechanism that's been proposed are through evolutionary constraints that might affect the fundamental mutational kernel, how mutations affect phenotypes, and then perhaps some type of a pulse might coincide with some type of constraint that's lifted, such as evolution on the gene regulatory network. Other mechanisms that have been proposed are also include population genetic dynamics, such as homogenizing gene flow with local adaptations among genes, inducing stasis, but bottlenecks and founder effects creating pulses. And what I'll focus mostly on today is regards stabilizing selection around an adaptive optimum to explain stasis and evolutionary pulses being explained by rapid adaptive shifts between optima. I just wanted to make a brief statement. One phenomenon that's typically not examined in these types of analyses are so-called macro mutations or mutations of large effect. But I just want to make a brief argument that that I think in some cases should be a valid or an important mechanism for describing stasis and pulses, because we do see for some phenotypes in mutation accumulation experiments that that mutations induce large effects on trait distribution. So here are 3 figures from a paper by Metzger et al., where they're studying gene expression evolution and induced mutations in cis and trans factors. And those mutations clearly are not normally distributed, which implies that they're not readily explained by small effect sizes. In any case, let me move on to sort of a macroevolutionary interpretation. This is concerning rapid adaptive shifts. And in the 80s, Mark Kirkpatrick and Russ Landy and others introduced some nice theory describing how population genetic theory and quantitative genetic theory can explain how populations shift between 2 optima. So on this y-axis, we have time. On the x-axis, we have a trait distribution for a population. And in a relatively short period of time, this theory shows that that a shift between these 2 peaks may complete. Using some fairly realistic biological parameters and using a theoretical model, Mark Kirkpatrick, for instance, showed that these transitions between 2 stationary optima can complete in say, on the order of 3,000 generations. Now, our fossil record, for the most part, is relatively sparse, but in a few cases, we have exquisite records of sequences of transitions between optima. This is a paper by Gene Hunt and others in 2008, where they recovered a fossil time series of 3-spine sticklebacks from Nevada. And in this fossil series, they found that adaptive shifts occurred again on the order of, say, 3,000 generations. Now, this number 3,000 doesn't mean anything inherently, but it's just to show and to lend evidence to the fact that these shifts can occur very rapidly, especially when you compare, say, 3,000 generations to something on the order of, say, 3 million generations. Now, one of the so far, I've basically been talking about descriptive models, quantitative models, and some fossil evidence in terms of how we hope to understand evolutionary pulses. But what's unfortunate is that for most living species, and most dead species for that matter, for most species, we don't have good direct evidence of when and where these pulses occur. And when it comes to studying evolutionary mode, phylogeneticists and macroevolutionary biologists have often turned to phylogenetic models to hope to estimate what mode underlies the traits that we observe today. So the rest of my talk is going to be focusing on how we apply these models in a phylogenetic context and what we can learn from them. So I'm going to give a very brief overview of some of the some phylogenetic models that I think most of us are familiar with, and for those who aren't, I'll give some cartoons and a quick overview of how these models appear in terms of the math. So here are three relatively commonly used models. We have a Brownian motion, an Ornstein-Uhlenbeck, and an early burst model from top to bottom. So the first model is a Brownian motion model that's typically described as a model of wandering change. And the idea is that you have some optimum that's changing slowly over time, and different lineages are attracted towards that optimum. I've colored the branches here only to tell them apart. They don't have any meaning apart from being able to identify independently evolving lineages. So Brownian motion is very simple. On the left-hand side, you have a trait which is a function of time. On the right-hand side, you have a Brownian motion, a unit Brownian motion, which accumulates variance proportional to the square root of time. And you have a diffusion rate parameter, sigma. And most of the stochastic processes we use for modeling trait evolution are essentially more complex versions of Brownian motion in one way or another. And what I mean by that is that Brownian motion is typically a special case of these other models. So an Ornstein-Uhlenbeck process, often this is meant to represent lineages that are constantly adapting towards a stationary optimum, or lineages who are adapting within, say, a fixed adaptive zone. And you can see that as you go from the left-hand side of the plot to the right-hand side of the plot, less variation separates the yellow, blue, and red taxa when compared to the Brownian motion model before. This model has a few more parameters. Now we have a attraction strength, how rapidly the lineage is attracted to an optimum. That's theta here. We see that there's this deterministic component that essentially is the distance of the process from the mean. And as a lineage, x of t, strays from an adaptive optimum, mu, the process will be drawn back towards that optimum. And that's the amount, the distance it's drawn is in proportion to the increment of time you're looking at. And then finally, on top of this deterministic process, we have this stochastic Brownian motion component. The third model, the third sort of simple model that I'll be talking about is this early burst model. This is a model that is meant to emulate the behavior of explosive or sometimes decelerating change, where the rate of trait evolution is most rapid near the formation of the clade, near that black dot on the left-hand side. And as we move towards the present, the rate of evolution decays. And the early burst model has a form that looks like this. For each moment of time, we have a Brownian motion that's creating this stochastic variation, but we're rescaling the rate of change in a deterministic sense. And this is through this function here, which is a gamma function, to borrow from Blomberg's work. The rate of variance decays with the rate of initial variance, sigma squared, decays with time, and that's what generates this that slowing or that explosive appearance in the slide I showed you previously. And one thing that can't be understated is that these three processes are absolutely wonderful to work with. I mean, for one thing, Brownian motion, Ornstein-Uhlenbeck, and early burst models all have a formulation that casts them as multivariate normal distributions. Normal distributions, or Gaussian, in this case, Gaussian processes, normal distributions are very wonderful to work with. They have this natural limit in terms of the sum of lots of small events with random effects will naturally induce something that's normally distributed. normal distributions have been studied in great detail in different fields, which means there's a rich theory to draw upon in terms of understanding how to basically manipulate these distributions to whatever ends you need. And the normal distributions also have a very simple probabilistic form, which makes it easy to work with in a probabilistic setting. And a feature, which I won't say is a good or a bad thing, but one feature of Gaussian processes is that they only are capable of generating continuous sample paths. And what that means is that if you assume that the parameters acting on the Gaussian process are constant with respect to time, you can't really induce features in trait evolutionary histories such as jumps or rapid bursts or things of that sort. In effect, this comes from the fact that very small probabilities are assigned to large amounts of variation in short periods of time. So most of what I'm setting up to talk about today are these models of pulse change, and to explore those models, I'll be relying on a class of processes known as Lévy processes. This is building on some work that I began to pursue in 2013 with Josh Schraber and Mason Liang. And Josh and I have been working on this quite a bit, even in the recent past. The basic idea behind applying Lévy processes to phylogenetic models is that these Lévy processes generate trait histories with jumps. And one of the contributions I feel that this paper made is that the fact that you have these jumps in the trait histories will complicate your ability to perform phylogenetic inference. And it means that to analyze these models, we need to develop some tricks to apply them to data sets. One feature that I mentioned is that these Lévy processes induce jumps in the trait histories. And so I want to give you an idea of what that looks like through simulations. These next few slides are basically going to be inspecting different aspects of these processes with respect to time. So suppose we have a Lévy process that's been operating over hundreds of thousands of generations. On the top, there's this pulse model. I'll explain what FR stands for in a moment. So on the bottom, we have a Brownian motion model, which is representative of wandering or gradualistic evolution. And so let's look at how these processes, the trait histories, change when we look at smaller and smaller time scales, and what impact does that have on the trait distribution we see at the present. So over 100,000 years, I'd say these two distributions are essentially not distinguishable from one another. If we look at, say, 50,000 generations, they're still relatively similar. When we start moving to, say, 20,000, 10,000 generations, 5,000 generations, in these shorter timescales, we see that at the top, we have this very pronounced peak and fat tails that we never seem to produce from the model below, the Brownian motion model. And for students of macroevolution, this is sort of pleasing because it means that we have a one-to-one correspondence between pattern and process. Here we have the process when nothing is happening or when the lineage is undergoing stasis. It generates peaks in our pattern of traits. And when pulses arise on these short time scales, we generate fat tails in our patterns of trait, in our distribution of traits. And this relationship of peaks and fat tails is often called positive excess kurtosis or leptokurtosis. And sometimes I'm just going to call it kurtosis for short. But what this means is that in a relatively short amount of time, you can produce a large amount of change. And this is the essence of what a pulse is. There are a few different types of Lévy processes that I'd like to introduce. This first one is called the jump normal process. And it is phenomenologically explaining a pattern of stasis followed by random pulses. How this is built is essentially you have a model x of t, or sorry, a process x of t, And it's a compound Poisson process, and the sum of a random number of independent effects. The number of jumps itself is a function of time. And what that means is that for short time intervals or low rates, you can actually have 0 jumps occurring over a finite amount of time. So this property is sometimes called finite activity. The effect sizes in this case are drawn, as the name suggests, from a normal distribution, but you can swap out different types of distributions for the effect size. It doesn't matter too much. In any case, this is a jump normal distribution. The next process I'll introduce is called the fluctuating rate model. This is a model with infinite activity. Something is always happening. And this reflects a macroevolutionary mode where you have perpetual evolutionary change with a fluctuating rate. And the way this model is composed is you have what's known as a time-changed Brownian motion, or stochastically time-changed Brownian motion. So our process x of t is a Brownian motion, but instead of having a simple constant value t, we instead have a random variable that's dependent on t and some other model parameters. And that model, that variable a of t is, in this case, going to be distributed by an inverse Gaussian distribution. You notice that the inverse Gaussian distribution is itself a function of time. Now, these time changes, there are different types of time changes. For example, for a Brownian motion model, there is no time change. We're saying that each unit of actual time on the x-axis corresponds to 1 unit of effective time on the y-axis. For you can have deterministic time changes. That's what we're doing for that early burst model that I described earlier. For each unit of actual time, we have a decreasing return on the number of units of effective time. So evolution will effectively slow with respect to time. And the fluctuating rate model that I just showed you, the blue band that is NIG. That's the actual name of the process. That should read FR. But in any case, I've basically simulated 5 time changes under that distribution for FR. This is what stochastic time changes look like.
Could you flip back to the model? Yeah, that one. Yeah. So that's interesting. So it's the inverse Gaussian that sort of gives you these bigger jumps.
Yes. So there are different types of processes that you can apply to. I'll show a figure that gets into some of the behaviors of different types of time changing functions in a little bit, but it's essentially the fact that for some increments of time, very little can happen, and for some increments of time, very much can happen. But for every increment of time, something has to happen for these infinite activity models. So, I'm not sure if you can see my mouse on the screen share.
I could there for a second. Maybe, yeah, I don't see it right now, though. Sorry.
Is it back? No. Okay. Yeah. But the idea is that for each increment of time, it's not necessarily the same size. It's a random quantity itself.
Right, exactly. Different than the Lévy model.
Yeah.
Cool.
And just like the jump normal model that I mentioned before, where you can have a different distribution for how large effect sizes are, there are various different fluctuating rate models or these time change models. This is the one I'll be working with for this talk, is this normal inverse Gaussian process. But there's also been some nice work done on these variance gamma processes. It's neither here nor there, but it's kind of fun to think about. They all look pretty much the same in my opinion. So here are 5 realizations from this stochastic time change. One question I'll touch on is that I'm asked often, how are these jump processes different from other stochastic processes? And some explanations are obvious and some are not so obvious. So let's see what this looks like. So first, let's start with constant rate Brownian motion models. There's some nice, as far as I'm aware, some of the first work actually describing these pulsed models of evolution from Hansen and Martins in 1996. And they mentioned some concern because when you have a model of Brownian motion, such as we have on the top here, it produces a particular variance-covariance structure. And in particular, the variance structure of that, the amount of variance you accumulate per unit time is proportional to the amount of time that you that the lineages have been evolving. And this is also true of these compound Poisson processes or these jump normal processes. They scale, the variance scales linearly with time. And if you look at this from the perspective of a multivariate normal, this is problematic because it suggests that you might not be able to identify these two different types of models. Now, it's important to recognize that the variance is a moment of a distribution, just like the mean is a moment of a distribution. And the kurtosis, the peaks and fat tails that I mentioned earlier before, that's yet another moment of a distribution. Now, when you look at only the second moment, the variance, it's true that Brownian motion models and Lévy processes, at least Lévy processes that have finite variance, tend to produce the same variance with respect to time. That is not true of the kurtosis. For Brownian motion models, Gaussian processes by definition have zero kurtosis, zero positive excess kurtosis. Now, these Lévy processes, such as the one that I'm showing here, this is a jump normal model. The kurtosis will decay the kurtosis as a function of time is equal to the kurtosis in a unit of time divided by the time itself. And so as time increases, we see that the kurtosis decreases also. And as time increases, eventually your model will look exactly like that Brownian motion model. Another question that I'm asked is, how do these pulsed models differ from models where you have a rate-shifted Brownian motion? These are sometimes called relaxed random walks, or the models that you can find in the base traits package. One way that people model Brownian motions is that they say for each branch in a phylogeny, we multiply the rate of change on that branch by some factor. So in this top part in black, we have a Brownian motion model where essentially we're time changing it, but we're time changing the Brownian motion by multiplying time by a factor r of theta. And what this means is that you can essentially pull that factor r of theta outside of the Brownian motion component, and you're left with a model. You're left with a distribution of traits that doesn't directly depend on time. And what this implies is that the kurtosis under these relaxed random walk models is constant regardless of how long a branch is. Now, for these pulsed models, as I mentioned previously, the kurtosis decays with time. And so one way that these two different types of processes differ is in how basically kurtosis is distributed with respect to time. And what that means for a phylogeny is how is kurtosis distributed with respect to divergence times among species. Another class of models that's very similar to these pulse models of evolution are called multi-peak OUs or multi-OUs. And how these models work is that you have an Ornstein-Uhlenbeck process, and the Ornstein-Uhlenbeck process is attracted to some adaptive optimum, but the adaptive optimum is not necessarily constant with respect to time. So here are some simulations I did where I have an optimum in black. It's very difficult to see the optimum in black because it's almost entirely coincident with a lineage with a large strength of attraction parameter alpha. So the blue and the black are almost perfectly coincident with one another. And as we relax the strength of alpha, say going from red to green to pink to orange, we see that the adaptive optimum is essentially less predictive of the actual trait that's evolving towards that optimum. So one relationship that these pulsed models have with multi-OU models is that as you increase alpha, we should expect that the multi-OU looks almost identical to the underlying process generating the shifts in the adaptive optimum. And so I can repeat that. I can do this. I can look at this in one way through, say, another simulation experiment, where I can simulate these kurtosis versus time curves under various processes. First, under black, which is the process describing the adaptive optimum. On the y-axis, I have the kurtosis, and on the x-axis, I have time. And just as we saw in these in this one realization, this one simulation, we see that blue and black are almost perfectly aligned. Here we also see that blue and black induce almost the exact same kurtosis with respect to time, because they track so closely. And as we weaken our strength of attraction, we see that the kurtosis becomes weaker and weaker. So I've focused on just a few Lévy processes here, but I want to emphasize they're just an enormous number of these processes, and they're interesting for different reasons. One Lévy process that one Lévy process that is has always been familiar to all of us is the Brownian motion model. That's a particular type of Brownian motion. It happens to be one without jumps. I introduced that jump normal process, but there are other compound Poisson processes with assorted jump kernels, such as jump kernels with skewness or excess kurtosis or features of that sort. The variance gamma process, normal inverse Gaussian process are more of these infinite activity processes. Stable processes and Cauchy processes are very fascinating processes that produce infinite variance. But one nice feature of all these processes is that some of the properties of Lévy processes allow you to basically compose new Lévy processes by adding together simpler Lévy processes. And these combinations of processes can allow you to produce models such as jump diffusion models. And so at the top here, I have this very simplified equation: x is equal to b plus j plus e. What this is saying is that we might want to construct some process x that's the sum of three different sources of randomness. This is sometimes known as a convolution. And here we have a Brownian motion model, a jump process, and some amount of interspecific variation. By summing these three things, we can essentially generate a jump diffusion process with some amount of evolutionary noise. And here I have the interspecific variation or evolutionary noise represented at the present with these distributions, but you can also visualize it in this manner, where it's essentially, if you were to sample a lineage at any point in time, it would be with subject to some noisy sampling process, or so if you were to sample an individual from a population, that's essentially evolutionary noise around a macroevolutionary optimum. Now the last part I'll talk about in terms of this very, very quick overview of Lévy processes and models of pulsed evolution is that there's been a great amount of work on this field over the past just about decade, I suppose. And while each of these studies has contributed something very valuable in its own right, I just want to highlight one feature of this. The first feature being that we can now analyze many more taxa than we could previously. And that's extremely important in a phylogenetic setting, because we rely on large species trees to generate the information necessary to fit these models. The second aspect is that when you look at the different approaches that people have relied on, a fundamental difficulty in studying Lévy processes in a phylogenetic context is that the timing and size of these pulses is not known, and somehow you need to integrate over all the possible placements and sizes of jumps or pulses. Now these I mentioned earlier that normal distributions are very easy to work with. And one reason they're easy to work with is you don't have to worry about this fact. With these jump processes, in red, I've highlighted methods that have, on one hand, greatly increased the number of taxa that we can analyze, but on the other hand, they generally rely on what's known as a sort of data augmentation method, where you're essentially trying to find and fit distributions of jump configurations that match the phylogeny. And this comes with some computational cost and complexity. An alternative way of trying to deal with the distribution of jumps in terms of their timing and placement and size is to attempt to integrate over all of those configurations analytically or numerically. The early work on this, unfortunately, in the early to mid 2000s, was restricted to relatively few taxa. The talk what I'll be speaking about for the remainder of this talk will introduce a method that Josh Schraber and I introduced, where we tried to circumvent the need for data augmentation, yet still allow our method be applicable to large numbers of taxa. So the reason that we were interested in applying these models to large numbers of taxa is because we wanted to get a sense of how common pulsed evolution is in nature. And to answer this, we needed to develop this new likelihood framework that's applicable to large data sets. And then we decided to test it against a large vertebrate body size data set. The key difficulty here is that we need a way to compute a new Lévy likelihood. I'm not going to get into very many details for how we do this, but I will sort of sketch the general idea. The idea is that we, just like everybody, we'd like very much to have a probability over traits that's easy to compute. And a product of independent probabilities is very easy to compute. The problem, though, is that our traits are not independently distributed. They share this underlying covariance structure due to the phylogeny. And in the 70s, Joe Felsenstein introduced a method for essentially erasing that phylogenetic covariance structure to induce a distribution of independent contrast probabilities. So how do we get from this starting problem to this endpoint? Well, what Joe realized is that we can prune off a terminal sister taxon pair, and that is an independent contrast with an independent probability from the remainder of the tree. But we can't simply just prune off the tree. We need to replace the pruned taxon pair with a pseudotip, which I've colored in blue here, plus some ancestral noise to account for the fact that we don't know exactly what that ancestral trait really is. This is noise that's sort of induced through the evolutionary process. And if we can do this once for a tree, we can do it as many times as we like. And finally, when we apply this recursively to the entire tree, we yield this distribution of independent contrasts. So that's wonderful. Now, what's unfortunate with the Lévy processes is that we can't use the simple tricks of storing ancestral noise in terms of simple variance. So the original method you could store the ancestral trait noise as a mean and a variance, but with these Lévy processes, because of the existence of kurtosis, we weren't able. Josh and I weren't able to find a way to do that so simply, and instead we developed a method that stores this pseudotip information using characteristic functions. And characteristic functions you can think of as an alternative representation. An alternative representation of a probability density function. The one issue, though, is that eventually we want to compute the probabilities of this distribution of contrast, which are all stored in terms of characteristic functions. So in order to compute that probability, we need to numerically restore our characteristic functions into probability densities using Fourier transforms. So we have a that's basically all I'm going to mention about the method for now, but that equipped us with a method that allowed us to test whether or not pulsed evolution is common or rare in a phylogenetic data set. And for this, we set out 3 criteria. First, is the process identifiable? Second, is the process frequently selected? And third, are the measurements realistic or not? So for the first point, is the process identifiable? To get at this question, we designed a simulation study. And part of our simulation study, in our simulation study, we wanted to account for the fact that tree error or phylogenetic estimation error could potentially induce what would spuriously be identified as a pulse. So on the left-hand side, we have a true tree on which we generated traits on. And on the right-hand side, we have a consensus tree taken from a distribution of supported trees. And if we change the topology and the relationships between taxa, suddenly taxon X2 and X4 are very closely related to one another, yet the amount of trait change shared between them is very large. And so tree error could induce what appears to be too much change for a young split, and that might be picked up as a pulse. Another issue that we ran into while we were composing our empirical data set is that for many data sets, the precision is actually quite low. And interestingly, when the precision is lower than a certain threshold, it means that it will induce this artifact of stasis, which means that certain taxon pairs can essentially exhibit no evolutionary change simply because of rounding error. And so this stasis is also something that could spuriously drive a signal for pulses. So throughout our simulation and our empirical analysis, what we did when treating our data set is we first ran a simple Brownian phylogenetic independent contrasts. We then corrected for precision error by dropping any clades with 0-valued contrasts. And secondly, we corrected for tree error by dropping any subclades with that were perceived as outliers. Or rather, we dropped the single clade that was the greatest outlier. Let me put it that way. This 33 of exempt, it means that in our empirical data set, we deemed that 3 of our 30, we dropped the outlying subclade for 63 out of 66 of our clades. 3 of them seemed like good, valid outliers. In any case, moving on to our simulation study, we designed a simulation study with sort of 4 quadrants here. We wanted to ask how well we could retrieve the true model when conducting inference under 4 settings. First, when we have a small tree with the true tree versus a small tree with a noisy tree or a tree with some amount of error, but also with the same with large trees. And in general, we found that for as long as the model parameters were relatively strong or characteristic of the underlying process, we found that our true positive rate was fairly high for pulsed models of evolution, which means that if we simulated under the jump normal model or a fluctuating rate model, we also identified one of those 2 models. You do notice that when we have few taxa, it can be hard to identify between those 2 cases, but that's a story for another day. We also noticed that under these simulation parameters, we have a relatively low false positive rate, meaning that if we simulate under a non-pulsed model, we do not accidentally identify a pulsed model, which means that our analyses should be relatively robust to those sources of error that I mentioned before. So first, I will conclude that the process is identifiable, at least in a simulation setting. Secondly, we are interested in whether or not the process is frequently selected in nature. For that, we compiled a data set of 66 clades across 8,000 species using a variety of phylogenies and body size measurements. The phylogenies are primarily molecular phylogenies calibrated using various means. Just to give you a very quick idea of what the data set looks like, We had 5 major groups: fish, amphibians, reptiles, birds, and mammals, with about 100, with over 100 species per clade on average. The trees themselves, the trees themselves were assembled either by collecting independent studies with independent tree estimates, or by pruning clades from larger supertree analyses. And the body size measurements were either in terms of length or mass, depending on what group of clades was studied. Just to give you a quick idea of how we were assessing model support. So we used AIC weights with the small sample size correction, and we applied a criterion that said a model class must be at least twice as good as any competing model class for it to be selected. So, for example, at the first row, we have stationary model. The pinkish color represents an OU process, and that received twice the weight as any competing class, so it was selected. On the bottom, we have a pulse model. Again, blue is twice as strong as any competing model, so that was selected. But in the middle, it's both the yellow, which is early burst model, and pink, which is the OU model. Neither is decisively greater than the other one, so we didn't declare either model as as having good explanatory power. We ran into because we had this threshold, it introduced sort of one tricky problem. We did in fact analyze our data set under a variety of pulse models of evolution, including jump diffusion models. And what that meant is often among all jump models, it was clear that the jump models were outperforming any of the competing models, but it wasn't always, but among the jump models, we couldn't always identify exactly which one was best. And because of this threshold, that would sometimes wash out our signal. While in fact, it's very reasonable to say that at least some pulse process was outperforming the competing processes. So in those cases, we took the jump process with the highest AIC support to represent the entire class, and then renormalized AIC accordingly. So to the results, when we applied these models to these 66 vertebrate clades, we found that pulse models were the most commonly supported mode, but pulse models are not the only mode, they're not the dominant mode of evolution in these phylogenetic analyses. If for a moment we withheld pulse models from our analysis, our macroevolutionary landscape across clades might look something like this, where the explosive mode, or an early burst model, seems to be receive the highest support. When we looked for interactions between the groups, such as fish, amphibians, reptiles, birds, and mammals with the different model classes, we did not find any strong relationships, which is in some sense reassuring, because it suggests that no single data set is driving the signal for pulses. From this, we can conclude that the process is frequently selected. It's certainly not rarely selected, which means pulsed evolution is at least something we should be aware of when talking about macroevolution in the context of phylogenetics. Finally, are the measurements realistic? To get at this, we were interested in asking whether our rate of pulses, it coincides with previous estimates for rates of pulses. And to do this, we defined a adaptive zone as an interval containing approximately 95% of the interspecific variation. This is that E parameter, that yellowish normal distribution that I showed earlier in the talk. And so we wanted to ask how often do adaptive shifts occur? Well, that's not simply the rate of jumps. That should be the rate of a shift outside of an adaptive zone. And so while we would expect that jumps of any size are relatively common, only jumps of a certain size or greater will occur at a lower rate. And so for all of the clades that selected pulse models, we compared the width of the adaptive zone on the x-axis and the waiting time between shifts on the y-axis. So on the lower left-hand corner, that would resemble a clade that had frequent shifts and narrow adaptive zones. On the right-hand side, we have a clade with rare shifts and wide adaptive zones. When we look at the median waiting time for adaptive shifts of this particular size of about 95% or greater, we get a median waiting time of roughly 26 million years. Of course, the median waiting time is going to depend on how you define how large a shift is. So you can imagine computing this waiting time for a variety of different adaptive zone widths or shift sizes. Another interesting thing is that these different Lévy processes will induce different waiting times for pulses of certain sizes. So on the right-hand side, I have 4 lines, the jump normal process fluctuating rate model. And then on the bottom 2, there are 2 jump diffusion processes, Brownian motion plus jump normal and Brownian motion plus fluctuating rate. And on the left-hand side, what you see is that these different processes will basically they have different waiting times until pulses of different sizes. And so an important distinguishing factor is that when you compare the 2 jump normal processes, JN, it can take a very long time on average to produce a very small amount of change. And this relates to that aspect of finite versus infinite activity that I mentioned earlier. Again, it's just it's sort of an interesting feature and something to think about for future studies. Now, when I mentioned that part of this was in order to make an estimate of how large these jumps are, to make a statement about whether or not our estimates are realistic. It turns out there's not very many estimates in the literature, but two that I could locate are one with Russ Landy, who using some empirically realistic parameters and a quantitative genetic model, said that adaptive rapid shifts can occur anywhere on the order of waiting times between shifts can occur anywhere from between 1 to 100 million years. Joseph Ujeda, in his blunderbuss paper, or million-year wait paper, had an estimate from the multi-burst model of about 25 million years. And you can see that depending on how wide we define our adaptive zone to be, we can produce shift waiting times that are within that ballpark. So this is reassuring. So somebody who studies macroevolution, being on the correct order of magnitude is a good place to be. But more can be done in the future, of course. So from this, we can conclude the measurements are realistic or at a minimum plausible. To summarize this small empirical study, I'd just like to say that this work introduced this phylogenetic method to compare the presence of pulses with respect to other macroevolutionary modes. We found that in simulation, pulsed evolution is detectable. We found that in empirical data sets, pulsed evolution is common and perhaps something we should be more sensitive to in the future. And just to introduce another sort of metric in the study of natural history is our study shows that these pulses arrive on the order of every 26 million years. And that suggests something about how often we might expect rapid evolutionary changes to occur in the diversity of life we see today. So I think I'm going to need to wrap up very quickly here. I'm not sure if the clock started late or not, but I'll wrap up quickly. I'd just like to touch back on some of these original ideas that motivated this talk, which is that those of us studying macroevolution are very interested in this final goal, testing models in nature. And in order to achieve that goal, we sort of have to work backwards. We need inference methods that let us apply certain models to nature. Well, what models what models are we fitting with these methods? They're statistical models. And where do those statistical models, they come from more, in the macroevolutionary context, they tend to come from descriptive models that have been discussed and studied under a variety of settings for centuries. And these descriptive models often are taking into account very diverse lines of evidence from evo-devo experiments to examining the fossil record. The difficulty, though, is that there's two real difficulties here. First is we have some serious challenges in terms of mapping these descriptive models onto statistical processes. I've sort of bandied about quite a few terms here without strictly adhering to what these terms mean. My personal experience working on this topic is that it's very difficult to talk about macroevolutionary processes without finding exceptions to those definitions. But in a statistical framework, you absolutely require strict definitions if you want to define the features of the process. So when I talk about a pulse or a burst or a shift or a jump, what exactly are we defining that to be? If we want to place gradual versus punctuated evolution at opposites of one another, or at least on a spectrum, at what point should the process be decreed gradual or punctuated, if at all? For these processes, you run into the issue of continuous versus discontinuous sample paths, and that's a mathematical feature, but that can be how to put this? That can echo some problems with invoking macromutations or saltationism with respect to older arguments concerning gradual evolution. And so it's another place that we need to be careful when talking about mapping biological onto mathematical models. And then finally, this coming up with good quantities to measure tempo mode and adaptive zones. I mean, in my opinion, there's still a lot of work to do there. Now, this process of mapping descriptive models onto statistical models, what I've been advocating, what my talk has basically done is shortcut the interspecific modeling step, going straight from a descriptive model to an interspecific one. And this is relying on arguments that say what happens on relatively short timescales might not seem to matter very much when examining processes on macroevolutionary timescales. But what you give up when you do that is you give up the extremely important and informative mechanisms that potentially will help us better identify different competing interspecific models of evolution. So if we knew something about, say, the genetic architecture of a trait, and we knew something about the population history of that of a lineage, that might give us a very strong idea of how a particular phenotype might emerge from that genetic architecture. But in contrast, if we simply say that traits evolved by a Brownian motion, so it's a model with a weak mechanism, it's fairly robust to assumptions, but at some point, we'll reach a limit in terms of what we can explain with those models. This is just a very brief overview of how the complications of these very simple models that we use, such as Brownian motion models, Ornstein-Uhlenbeck models, and so on. These simple models are often purposed to explain different phenomena depending on what scale you're working with. So a Brownian motion model on a microevolutionary scale might be consistent with neutral evolution by genetic drift. On a macroevolutionary scale, we as biologists are often still invoking a strong adaptive explanation for how those traits are changing. And so there's a great amount of care that needs to be taken when talking about these processes across scales, but also how at some point we're going to run into trouble in terms of identifying a Brownian motion that we say is describing a macroevolutionary process versus a Brownian motion that we say is describing a microevolutionary process. We need more to actually distinguish those two scales properly. So another thing I'd like to touch on is that different when we talk about evolutionary mode, depending on who you speak to, it's going to be on a different evolutionary scale. When Gene Hunt's work, when he's talking about evolutionary mode, is often within a single fossil lineage or chronospecies. When in Joseph Ugeda's work on the multi-burst model, when he was looking across essentially a large group of metazoans, that's an evolutionary mode that spans lineages. Somehow we need, I think an interesting way forward in phylogenetic work is to talk about how we can induce shifts in modes on phylogenies. So, for example, just as has been done with Bayes traits, Atour and Bayou, where you have shifts in parameters as part of the evolutionary process, you could also imagine shifts in evolutionary modes or shifts in evolutionary processes with respect to branches and clades. Another way that the scope of a mode might be constrained is across traits. One thing that Josh had touched on is that with these gene expression data sets, we're often now applying a single evolutionary mode to all of our traits at once. So all of our traits evolve on, all of our genes expression levels evolve under a Brownian motion, or all of them evolve under an Ornstein-Uhlenbeck. But different traits might evolve according to different modes, depending on, in the example of gene expression, depending on the underlying genetic architecture. So there's room for us to talk about how modes might vary across different traits and across species. And here you can imagine using these Dirichlet process priors or reversible jump methods to get at that. Even if you don't really care that much about the evolution itself, Joseph Ugeda and others made a really nice point about, despite whether or not you care about the phylogenetic, the model underlying the phylogeny, if you want to compare biological traits between species, you need to somehow be at least aware that the phylogeny is inducing correlations of different types. And in their recent work, they pointed out that rare events of large effect can induce spurious correlations between random variables if you assume a simple Brownian motion model for phylogenetic independent contrasts. And so this is a place where I see that these Lévy processes and associated models could be potentially very useful for not causing these rare events of large effect to induce, to act as outliers from the perspective of a Brownian motion. I think I'm more or less done. The last thing I just wanted to touch on is that the fact that every time we find a new model, there's the macroevolutionary spectrum as it's applied to different lineages or clades or traits or what have you seems to change. And I'm really looking forward to see how this changes in the future. Just because we find the signal for Lévy processes today does not mean that the story ends there by any means. I think the next direction that will be exciting to investigate is not just simply trying to emulate this interplay between stasis and variation, or stasis and pulses, but rather what processes are actually creating this stasis and generating this variation. And in recent, there's been just an enormous amount of recent work I've enjoyed following, talking about how we can actually bridge mechanistically microevolutionary and macroevolutionary processes. Okay, I think that'll wrap it up. I'm not exactly sure where I am on the clock, and I hope I didn't delay anybody too much, but I'd like to thank everybody for listening, and especially Eric again for giving me this great opportunity. Thanks.
Nice talk. That was really beautiful. I liked how you sort of went from the big picture to some specifics and then back to some like larger themes. So we will take questions. You can type them into the chat box in YouTube or tweet them. So, I mean, going back to your sort of data-driven study, I mean, you said, okay, well, if we look at like, are the fish having more pulsed evolution than other things or something like that? You didn't see a signal. I mean, is there some other classification that you could do that you think would be sort of more promising for finding some characterization of these different evolutionary patterns by some other covariate?
Yeah, you know that, yes. So one thing that came up in review, I think there are lots of other sort of extrinsic features of clades that you could potentially covary with model selection. And one thing that came up during review for that particular paper with the vertebrate evolution was, do different aspects of the tree itself that the trait is evolving on, different aspects of the tree tend to induce or tend to be associated with different models of evolution. And when we looked at features such as number of taxa or clade age, or there's a gamma statistic that is essentially called tree gravity, which explains how or describes how tippy a tree is. We didn't find much in terms of the phylogeny that described that that pattern. You can imagine looking at other things though, like I could imagine that fish or pelagic things face very different types of geographical constraints relative to terrestrial organisms, and that might affect the way that an evolutionary landscape looks to them. So yeah, you can definitely imagine looking at different features, but just at a gross scale, looking at those 5 major groups, we weren't able to find any relationship.
Right. Well, I mean, in a sense, it would be neat if one could, try to be a little bit, like, I can't resist but say like some sort of more machine learning thing where you're, where even like your models are getting a little bit more flexible, and instead of sort of like you were saying, okay, well, what's, we don't know what's coming next, but maybe there's some more non-parametric thing or something that we could use that would step back and be sort of a more general meta model.
Yeah. I've been really enjoying watching how machine learning is beginning to grow up and mature in the phylogenetic literature. I know there's been a lot of work with machine learning and population genetics, and one of the things that's challenging about that is with machine learning methods, you don't always. It can be yeah, how to put this? You have one replicate of biodiversity on Earth, right? And
Yeah.
many of these machine learning methods require that you can say withhold some data and study and basically train your data set to a subset of data and use it to predict the remaining data set. And with these phylogenetic studies, when you're talking, we can't go out and say sample it from nature another group of vertebrate radiation, right? And so in order to actually apply these machine learning data sets, it seems that you typically have to resort to constructing sort of simulated data sets that that hopefully capture the behavior that you want to model. And because of that, because you basically have to simulate these data sets, presumably under some model, there is some risk in sort of basically getting out what you put into it, if that makes sense. Because we don't know the underlying macroevolutionary model, if you want to sort of generate these training data sets, it seems like you potentially have to explore a very large space of underlying macroevolutionary models just to generate simulated training data sets. And that's not to say that that's good or bad. I mean, it's probably the fact that there are lots of evolutionary processes and we can't actually identify them, and that's just the world we live in, and tough luck. But it is I can see that there are some fundamental challenges to applying the machine learning stuff, although I'd like to see or hear what other people think about that.
Awesome. Well, I actually do have to run, and we are a little bit over time, so I'm going to stop here, but say thank you very much. And
yeah, thank you.
Yeah, thanks again.
Bye. Take care.